I think another big area where actually our research might dovetail with yours is this whole concept of engineering new tissues and organs. That's actually led from work in our own lab to, artificial skin for burn victims, to new blood vessels that actually the FDA approved that actually are being used to treat soldiers in the Ukraine.
I'm Insoo Hyun, Director of the Center for Life Sciences at the Museum of Science. Today, my guest is Dr. Robert Langer, one of the founding figures in bioengineering. He's one of the world's most cited scientists, and he's helped start more than 40 different companies, including Moderna. Today, we talked about the future of medicine and the role of engineering in healthcare. Well, Dr. Langer, Bob, it's so great to have you here on the program. I'm really glad you can make time for us.
Oh, well, thank you for inviting me. It's a pleasure.
Great. So your lab at MIT is really one of the largest in the world. At any given time, you've got about 100 researchers, well over $10 million in research funding. So you've been very, very well established in biotechnology and science for a very long time. You've had a very long and distinguished career. I want to ask you, before we get to the things that you've achieved and all your wonderful accomplishments, I wanted to ask you, what are some of your biggest failures?
Well, I have a lot. I mean, I probably failed way more than I succeeded. I think a lot of people in science do. Well, I mean, the first set of failures is I couldn't get a job. I tried to get several times. I couldn't get a job. I started out in 1974, and I'm a chemical engineer. I got a lot of offers from oil companies, but I wasn't excited about doing that. So I wanted to get it. I had helped start a high school for low-income students and got very involved in math and science curricula and in chemistry curricula in particular. And I would write colleges to do something like that because there were assistant professor positions available to do that.
So you wanted to get your foot in the door doing some teaching locally, and you weren't really getting any callbacks?
That's correct. Nobody would get me back. And it wasn't just teaching. What I was really interested in, and that's one of the things I did at that school, was try to create new curricula to make chemistry and math more exciting. And in fact, the first year I remember teaching, they had 37 students in the school, and I think 5 signed up for math. It was a very liberal school. But the second year, 45 out of 50 did. And then we also, MIT gave us some funding to start a chemistry curriculum, and the students really liked it. It was kind of very practical chemistry and things like that. So I was interested in doing something like that, but I wrote to maybe 40 different colleges, and I don't know that any wrote me back. So then I had the next idea was that I could do something in the medical area because I wanted to help people. And I wrote to a lot of hospitals and medical schools, and they didn't write me back either. So I had lots of failures. And then I ended up working at Boston Children's Hospital, and I thought I was doing some pretty good research there. But still, I liked it very much. But then after a couple of years, I applied for faculty positions. Now I had a bunch of ideas about how I could use chemical engineering, which was my background, to help people. So I wrote to a lot of hospitals and medical schools and chemical engineering departments. And nobody would write me back. I didn't get any jobs. Finally, I got one in the nutrition department. So I failed over and over again. And then I got my first 9 grants rejected because people didn't think the work that I'd done, which would ultimately lead to a lot of the nanoparticles that deliver large molecules that you hear about today. I mean, they didn't think it was right or make sense. I mean, I had, trying to even get that to work, I think I failed 200 times before I finally got it to work.
I mean, with all these failures and slow starts, I mean, how do you keep going? I think a lot of people would give up after a little while. And there weren't probably a lot of people kind of like you at that time, kind of doing that kind of thing or wanting to do the same kind of thing, right? There wasn't like a big cohort.
Yeah. No, that's true. I was when I worked in the hospital, I was the only engineer there. But I think what keeps you going, well, first is the alternative. The major alternative I started thinking about was taking some time off and working in an ice cream parlor because I always like ice cream. I think what keeps you going is not only, like I say, the alternative, but I believed in what I was doing. And I also made a commitment to Dr. Folkman, who was my boss, I wanted to solve these problems because I thought they were very important. But it was depressing. I mean, it was depressing to not get grants, to, get rejected and stuff like that.
One of the most famous companies that you helped found was Moderna.
Yeah.
And tell me a little bit about that. I mean, about the origin stories of Moderna and why that sort of became such a huge success.
Well, for me, I mean, it goes way before Moderna in the sense that when I got done with my PhD, this goes back to 1974. As I mentioned, I worked at Boston Children's Hospital, and really the project I began working on, which involved studying blood vessel growth, but there was no way to really study it. So what I had to develop was a way to deliver large molecules, which large molecules are things like proteins or nucleic acids like RNA or DNA. And I had to create at least tiny little particles that I could put in places that could deliver these molecules. Nobody had ever done that before, and actually people said it was impossible. I spent several years working on this, and I always tell the story. I found 200 different ways to get it to not work. But eventually I did, and we published this in Nature in 1976, and that was the first time that there was a delivery system for nucleic acids. Anyhow, we kept working on it over time. Sometimes companies would use it. Sometimes I even got involved in companies using it. And of course, myself and lots of other people over the years would reproduce it, make it better, and so forth. And our own lab solved different problems over the years. We were one of the groups that came up with the idea of adding polyethylene glycol, which is something in Miralax that people take, but that was important to prevent, to make it last longer and to make it not stick together. And we came up with Dan Anderson, who was in my lab then, now he's a full professor at MIT, came up with this way of making thousands of different types of lipids. And Rohit Karnik, who was in my lab, also now a full professor at MIT, came up with ways of manufacturing these things. So we made various contributions. Anyhow, in 2010, Derek Rossi, who was a professor at Harvard, he basically came up with a way of using messenger RNA to actually, it goes to your area. He was using messenger RNA to convert a regular cell into an iPS cell. Before that, Yamanaka had done that with viral vectors, won the Nobel Prize for that. But Derek did it with messenger RNA. And he was interested in possibly starting a company on that. He went to different venture groups, and they didn't give him any money. In fact, he wasn't the first one to do that. Actually, Kariko and Weissman, who made a major discovery, won the Nobel Prize for modifying messenger RNA, also did that. And it was very hard, I think, to get funding. Anyhow, he came to see me. He mentioned three or four different ideas that he was interested in. One where it had to do with kits. Another did have to do with like kind of more tissue engineering with the iPS cells, and another was proteins. I thought the protein idea could work partly because of the nanoparticles that we'd done, and partly it just, would make sense. But the key for the nanoparticles is that would be a way to put messenger RNA in a nanoparticle, give it to a patient, say by injection, then the body would do everything. And basically the delivery problem is easier. If you use a viral vector, you have to get to the nucleus. Here you just have to get inside the cell, like into the cytoplasm. So it seemed to me that that could be a pretty powerful idea, and you could do everything much more quickly. You wouldn't need to, say, pick eggs for something like that, grow them up for a year or two to guess at what type of vaccine you're going to make or what kind of molecule you're trying to make. So at any rate, so Derek and I talked, and then we talked to Ken Chen, who was a cardiologist, and Nubar Fayan, who started Flagship Ventures. I had the fortune of being on Nubar's thesis committee when he was a graduate student at MIT. So the four of us started it, and then Nubar said to me one day, he said, well, did I ever meet Stéphane Bancel, who was the CEO of a big French company? And I had, and I thought he was very impressive. And Nubar said, well, I think maybe we could get him to come. And so he came, and then we got Stephen Hogue, Melissa Moore, some other just outstanding people. And what happened was by 2019, Moderna had something like 13 products, including, some vaccines in clinical trials. And then, of course, COVID hit. And but Moderna was really, I think, uniquely positioned to solve that problem because, we had by this time a big plant in Norwood that we were going to use and still will use to make personalized cancer treatments. But at any rate, that's how it got started, and it was really quite remarkable that when that happened, we were able to design the vaccine in a matter of minutes, whereas before it would have taken years, and get the first clinical trial, first patient treated just 2 months after the whole thing started.
Yeah. Now, some people in our audience may not quite understand how mRNA vaccines work, for example, against COVID. Can you just give a quick explanation of how they actually work?
Sure. So a way to think about vaccines at a certain level is DNA makes RNA makes protein. And certainly a lot of vaccines, the way they work is because you have a protein. And the body then makes, and if it's foreign, the body makes antibodies and you generate immune cells that will attack anything that protein is on, basically. And that's basically what happens. You have, if you take DNA makes RNA makes protein, you put the RNA in a nanoparticle, you inject it in the body, it gets into the cells, and it makes the protein. And then because it makes the protein, then you have these immune cells and antibodies that if it sees a particular protein that it's foreign, it will go after it. Well, in this case of COVID, you have something called the spike protein that's attached to the COVID-19 virus. So what happens is when the immune cells and antibodies see that, they go after it and destroy it. And so you get, so either you will not get sick, or if you got sick, you'd get a lot less sick. And I guess the estimates have been that it saved 20 million lives. And so it had a giant effect, but it also, I think, prevented hundreds of millions of hospitalizations from what I understand. Given
the technology of mRNA vaccines, this platform makes it possible then to develop vaccines very quickly, right?
Yes.
For novel uses. Do you think there'll ever be an mRNA vaccine for like the common cold or flu?
Well, flu definitely. That's being worked on. In fact, the data out of Moderna, which has been published, is certainly the most effective vaccine for flu, much better than the standard vaccines. And you'd expect that in a way, partly because you're not going to be guessing either, about what.
So you could design that mRNA just exactly to like what you need to create the proteins.
Yes, that's correct. And so, so I think for flu, like I say, that's already done. I hope that it will be used in the near future. I mean, right now it's hard to know what's going on in terms of regulatory issues on vaccines, but I certainly hope. I mean, in terms of saving people's lives, it will. I mean, if it gets used, I think the common cold, that may be a different bar. I mean, I think you have to, and again, this goes certainly beyond where. I'm certainly not a policy expert, but I would say that that probably is more complicated. And then the question is the juice worth the squeeze in that case too? But I mean, so that's at least how I'd look at it. But anything is certainly possible.
Could there be a HIV vaccine?
Well, that's been worked on too. Absolutely, yeah. I mean. People are at Moderna and other places are working on that.
What do you think?
We're working on it in our own lab with the Gates Foundation and with Dan Baruch at Beth Israel Hospital.
What do you think are some pretty common misconceptions about mRNA vaccines, and why do you think people have some of these misconceptions?
I think people have misconceptions because the people that sometimes talk about them don't necessarily know. I mean, either a lot of it seems to me fueled by, I don't want to say the media, but it's fueled by, misinformation that is often could be on websites. I mean, I suppose the other thing is there's a whole group of people called anti-vaxxers, and they're now, because of, say, Bobby Kennedy appointing them, are in significant roles. So I think that it really has to do, a lot of it to me is a communication issue. I mean, but again, I'm no expert on it.
Yeah. So you told me that you don't actually have a whole lot of federal government funding for your work these days, but in spite of that, maybe it's not true in your situation or for your lab, but do you think the future of mRNA vaccine development in the U.S. is a little bit under jeopardy?
I think under jeopardy for a short period of time. It's hard for me to believe in the end, throughout history, certain things, the Earth rotating around the sun, other concepts, certain times science can be in jeopardy. I mean, it's the only country in the world that has this issue right now. Nature, which is a prominent journal, certainly pointed that out. It stands alone in terms of those kinds of issues, according to Nature and the writers there. But what I'd say is that I usually find these things to be short term. I mean, and I don't believe that in the end. I mean, I think science always wins.
Yeah. But you've been a leader in biotechnology and you've been in the field for decades. I mean, looking back over the last several decades, what to you were some of the key scientific pivot points or revolutions in the life sciences that you really think people should know and about and celebrate?
Well, I think there are a lot. I mean, it depends how you look at it. I think there's been a lot of progress. I mean, starting with things on getting a structure of DNA and RNA and things like that. But then I think, really better understanding molecular biology, biochemistry, cell biology, discovery of new kinds of RNA like siRNA, messenger RNA, discoveries of gene editing kinds of approaches like CRISPR certainly would be important. So those in the biology arena, I think those have been some of the things that have been important.
Yeah, and looking ahead, I mean, what do you think are some of the key problems that really, to you, should take priority for people to solve scientifically?
Boy, I wouldn't know where to start. I mean, I think there's so many, both at a fundamental level and at an applied level. I'd say, really trying to understand abnormal cell growth like cancer and things like that. I think certainly understanding the brain, I think, is a giant area. And then I think applying those things to different treatments. Can we come up with better treatments for cancer? Can we come up with better treatments for diseases like Alzheimer's or ALS or Parkinson's? But I think it's pretty unlimited.
Yeah. In your opinion, what are some of the key engineering problems in medicine?
Well, I still think delivery, most people would say, is a big problem. And in that area, I'd pick a couple of things. One is targeting to specific cells. I mean, could you target to cancer cells? Could you target to, any specific cell in the body to treat disease? Another one would be passing through certain barriers. Could you take these molecules orally? Could you cross the blood-brain barrier? But those are just some. I think another big area where actually our research might dovetail with yours is this whole concept of engineering new tissues and organs. And that's actually led from work in our own lab to, artificial skin for burn victims, to new blood vessels that actually the FDA approved that actually are being used to treat soldiers in the Ukraine. But it also, and hopefully many other things too, but it also leads to the possibility of organs and tissues on a chip, which may tremendously accelerate drug testing. In fact, the FDA has recently talked about the idea of really using them much more than say animals and so forth. So I think that those are also big areas that engineers can play a big role in.
Yeah, an organ on a chip system is where you take human stem cells, you create specialized cells of interest like liver cells or airway cells. You put them on an inorganic chip, microfluidic little chip system that kind of helps you study how drugs interact with these cells in a kind of lifelike, human-like way. That could also potentially be an alternative for testing some drugs in animals, for example. So for the organ on a chip systems, for all those engineering systems, you talked about delivery as being kind of a key engineering problem. I mean, one of the really interesting things you did early on was develop a drug delivery system for brain cancer. Can you tell me a little bit about that? Like, what was that system? How did it work?
Yeah. Well, I'll take a step back. When I was working at the hospital, one of the things I noticed was that almost all of the materials that were used in medicine weren't really designed for medicine. They would be some medical doctors going to their house and finding an organ or tissue, and finding an object in their house that kind of resembled the organ or tissue they wanted to fix. So the materials in the artificial heart, that's a lady's girdle because it has a good flex life. The material in a breast implant, one of those is a mattress stuffing. And I thought, well, we could make materials.
Maybe do a little better than that.
Yeah, have a different strategy and a different paradigm. Yeah. So I started doing that back around 1979, 1980, and we made a family of polymers that would dissolve in a much better way. In fact, at that time, there were hardly any degradable materials that you could use in people or synthetic polymers. So what happened in the case of the brain implant and gliadel, which is now still used almost 30 years later in many countries, is that Henry Brem, he was a young neurosurgeon at Johns Hopkins, actually went to Harvard Medical School. And he and I worked together starting in 1974 when he was in Dr. Folkman's lab. And he came to see me one day and said, because he was treating brain cancer patients, and he said, well, could you ever come up with a way that when he operates on a patient, he could leave something behind that could do local delivery to destroy the remaining tumor. And since we had developed these materials that dissolved in a much better way, would be unlikely to dump a cancer drug, which might be toxic, we tried to put certain cancer drugs like BCNU, it's also called carmustine, in little wafers made of these materials. And, we worked, first did studies in animals and showing it's safe, animals showing it's effective. And we wrote a patent, the company licensed it, and ultimately the FDA approved it in 1996. It's been used in well over 10 million patients in over 30 countries, and hopefully has really improved lives. I still think it's not only important in its own right, it's kind of a paradigm that you could do local chemotherapy rather than systemic chemotherapy. So you get high concentrations in the location where you want, but low concentrations in the rest of the body where it could cause harm.
That's fascinating. So I've talked to some chemists who work for drug companies, and they're often tasked with coming up with slow release molecules or things that kind of like let the drug sort of infuse in the body in the right dosage, kind of over the right time course. But you're talking about actually engineering that kind of does similar types of work. That's fascinating. So when you start ideating or thinking about new engineering problems or approaches to medicine, do you ever sort of self-impose a design constraint for your engineering task that says something like, whatever we come up with, can we try to make it more feasible that this product or this innovation could be deployed in resource poor settings or areas that kind of don't quite have like the setup that we have in Cambridge?
Yeah. Well, I mean, the answer to that's yes. And let me also take a step back on that. But the first thing, whenever you come up with an idea, like a new particle or or this idea of delivering nucleic acids or stopping blood vessels to treat cancer, a lot of times people will tell you that it's impossible, it will never work. So the first thing you have to do is prove that it can and establish some principles to enable it to work. Irregardless, I think, of the resource setting, because you have to make a discovery or invent a material that doesn't really exist and that people think won't exist. And so that's the first step. I think once you do that, then you're in a position to start to do what you said. So so Bill Gates came to see me back around 2012. I'd say right now over half our lab is devoted to exactly the issue that you mentioned. And they've been very, very helpful. We've started actually with their help, I think, over 20 clinical trials on different aspects of this. But some of the things that are cost, some of the things are pills that might last a lot longer than a day, that might last a month or maybe even a year for the entire course of treatment.
Wow.
Some of the things are vaccines. When you talked about vaccines, I mean, one of the big issues is a lot of times you have to do repeat injections. We're working on creating a vaccine where you'd never have to do that. And then also nutrition. I think each problem or each issue in the developing world, you have to look at things on a case-by-case basis. So when we work with the Gates Foundation, we build on the principles that we've developed in the lab in terms of proving things are possible and understanding how to do it. But to the point you raised, the way they always talk about it is a TPP, a target product profile. And some of the things that are important in the settings that you mentioned are lower cost, higher, being able to sustain better shelf life, being able to withstand, very warm temperatures, in shipping and things like that. So yeah, we do that on quite a few things. So we probably, like I say, over half our lab is working on that. And we've also spun off some companies that are working on it with the foundation helping us.
Yeah, one of the things you helped develop were these transdermal release systems. People must still be working on that, right? I've heard of people working on patches where it doesn't require a syringe to deliver, a drug, but you can just kind of put a patch on and it sort of gets in there.
Yeah, that's right. We've been involved in that for a number of years. Some of the things that and those are used for different treating, heart disease like nitroglycerin, motion sickness. For scopolamine. There's a number of areas where people use patches, but they're limited because what you can do because your outermost layer of the skin, the stratum corneum, that's almost like a brick wall. In fact, if you look at the structure of it, it looks like something like that. But anyhow, we've been working on different things. One of the things we've been working on, which is a type of patch, are microneedles. We've also looked at how forces like ultrasound or electricity could, get molecules through the skin. So, those are all things that we've worked on in our lab. And there are certainly many transdermal products, but I hope that some of the newer things that will be useful in the future would be these micro needle patches. In fact, to the end that you mentioned before on things in the developing world, Anna Jakulenik, who works with me, BARDA, in the U.S. government approached her about could we make a simple portable 3D printer? A lot of these are real big. Could we make a tiny one that could be shipped anywhere in the world, in the United States, Africa, wherever, remote areas, where you could make these 3D printers and even put, things like RNA in them and so forth. So we work on quite a few different things, or at least try to create the principles for them.
Tell me about your work around diabetes. Do you think that this is more of an engineering problem or a biology problem? What kind of problem is it that we need to solve?
I think it's both. I think, of course, a lot of these things are both that we've talked about are both biology and engineering problems. So we are doing a lot on different parts of treating diabetes. One, of course, is an artificial pancreas. We've had help from Juvenile Diabetes Foundation and now T1D and the Helmsley Foundation and Dan Anderson, who was a postdoc with me. We started this a number of years ago, where you can encapsulate islet cells, and that actually can protect them from being destroyed. So you could inject them. But then a problem that occurs is the things that you encapsulate them with themselves get encapsulated with what's called fibrous tissue. And we've created some materials that seem to be able to stop that. And so I think that will be very, very important. Another issue is getting enough oxygen inside the system, and we've been working on that too. So we have started one company that now Eli Lilly acquired to address the biocompatibility issue, with the fibrous encapsulation issue. But we're starting another one now that's going to maybe incorporate some of those things as well as the oxygen issue to hopefully come up with an injection that could create an artificial pancreas. But we've also done other things. Right now, little kids, have to get injections of insulin, and that actually delays treatment sometimes for a very long time, years. We've come up with a way to deliver insulin orally. It's kind of a little pill that you can swallow, and we've actually tested it in animals, and it's not just insulin that could be delivered. You could deliver messenger RNA, or you could deliver antibodies. But basically, you can swallow it, and it at least in animals, it works as well as swallowing it as just injecting it. And it seems to be quite safe. And we've even, with our collaborators, have done 65 patients where we've just looked at the little system and it hasn't caused any harm. We haven't been putting drugs in it. We've just been looking at safety.
That's interesting. So when you ingest something, though, the liver, it has to go through the liver. But I take it when you inject a drug, it doesn't. You're not really competing with the liver. The liver doesn't get involved for injectables, but it does when you.
Well, that's a good question. So what we did in this case is our little pill injects something in the stomach.
Oh.
So I work with a terrific gastroenterologist, and he's a mechanical engineer now too, Gio Traverso. And he came to my lab as a fellow, and now he's a faculty member at MIT. He's done incredible. And what happens is that I probably even have one of those pills somewhere on me, but basically we modeled this. You swallow it, and we actually modeled it after what's called a leopard tortoise, and it has a weight distribution, so it tumbles after you swallow it right into the stomach. And then what happens when it gets into the stomach, it just turns the right way and injects it in the stomach, so it goes into the bloodstream like to the same level. And it doesn't hurt the stomach. I mean, it's a very tiny injection, and the stomach actually, as Dr. Traverso says, it responds and heals almost immediately.
I've never heard of that. So something that you can ingest, but it gives you a shot.
Yeah. Well, there's a few places that are doing that, but we've actually published that in Science, one of the and Nature and Nature Biotechnology, and then one of the Cell journals. But, like I say, now we're starting a company on that too, because I want to see it get to patients.
Wow. That's fascinating. So, just engineering solutions for medicine. In the next 20 years, what do you hope medicine looks like?
Well, I just hope we have better treatments for cancer. I think there's a good chance of that. One of those things that Moderna has worked on are personalized cancer treatments, where rather than design a vaccine to go after, say, the spike protein, which I mentioned, you could look at what the mutations are on cancer cells, and then design something that will make an mRNA that will stimulate the immune system and form antibodies that will attack anything that those mutations are on, and then destroy the cancer. I mean, that's one thing. And people are also coming up with better antibodies. And engineered cells like CAR T cells to treat cancer. So I hope we'll have better treatments. I also hope we'll have better treatments for, brain diseases, which is certainly, something very, very prevalent. I think there's been some progress in heart disease, but I'd like to see better treatments there too. And I think we will. I think all those things will happen. But, we'll have to see.
AI has been all over the news lately, and I'm wondering if, in your opinion, AI will have a very big impact in the kind of work that you do.
I think it. I mean, the answer is yes, but it's not new. I mean, AI has been around for about 80 years. In fact, one of the things that I did with Nick Galakatos and Michael Sema and a few others was start a company about 26 years ago with some of my other students, Dave Putnam and Hongming Chen, which we call Transform Pharmaceuticals. And we just looked at a simple thing, that 50% of the drugs that people take orally, small molecule drugs, aren't even soluble. So, and at that time, what people were doing was basically, maybe they try adding different substances that they thought were safe one at a time. So, and we thought, well, why can't we do this in a high throughput way, test thousands of excipients and combinations, use what we call machine learning to look at the structures. Maybe 5 out of 1,000 would look good, look at the structures that work well. And that would then give us a second group that we'd pick. And then maybe we'd see the solubility rate got higher and higher. And that ended up being very, very successful. We actually, got a number of products, and we actually also used this high throughput approach to come up with new what are called polymorphs, crystal structures, which might be more stable. And that was actually important because there was a drug called ritonavir in 1996, an AIDS drug, that all of a sudden changed from polymorph 1 to polymorph 2, and it behaved differently, so the FDA made them take it off the market. And what Transform did is, 7 years later, just in 2 weeks, they found polymorph 1, polymorph 2, and 3 polymorphs that had never been discovered before. So this goes back 25, 26 years where we used a lot of these techniques and made a big difference. I mean, Trent Johnson, that's now part of Johnson & Johnson. At any rate, today, Dan Anderson and myself, Gio Traverso, Anna Jakulenik, the people that work with me, I mean, there's a lot of ways we use AI. I mean, we're using it to, on the one hand, make better lipid nanoparticles. There's thousands and thousands of lipids that we're looking at, and in silico, which is like sort of an AI kind of thing, you can make millions. And so we're coming up with better and better lipids. We're also even using it in the nutrition area, where we're designing better systems. In fact, the Nobel Prize this year was for these metal organic substrates and complexes. And we've actually, with Anna and some of the people there at MIT in our group, we've come up with designs that can give iron better, really, for the developing world. And what's kind of cute is that a couple of my students, that did this, they actually put it in coffee. And and when I give Bill Gates a lecture once a year, and so when we tasted the coffee, nobody could tell the difference between their coffee and Starbucks coffee, and also they won the Inventor of the Year for the best invention, the two students.
Wow! Looking back, you've done so much, you've achieved so much. What's your proudest moment?
I don't know that it's a particular moment. I'm always incredibly proud of how well my students and postdocs have done. I think over 1,000 of them have started companies or are, and many, started companies or professors, I think about half and half, all over the world. I think 10 of my students are professors at MIT, I think 11 at Harvard, 11 at Stanford, but they're everywhere. And they've done great. I think over 60 of them are in, maybe it's over 70 in different national academies. And, so I feel very proud of how they've done. I mean, that's really one of the main things I do is hopefully train people who are doing good things for the world, and they train others.
Well, Bob, it is an incredible body of work. Thank you for all you've done for science and medicine, and thank you for joining us today at the Big Question.
Well, thank you. It's an honor to be here, really. Thank you.